Welcome to the Breakthrough Broadcast, a podcast about innovative STEM solutions to real-world problems. From emerging technologies to practical research, Drew Reck explores how science, engineering, and bold thinking are shaping the future.
Hi, I'm Drew, and today we're exploring the first episode of our first two-part series. A series about one of the most transformative tools in modern genetic science, CRISPR. This technology has completely changed the way we approach genetics. Not long ago, editing DNA was slow, expensive, and uncertain. Scientists had to rely on clunky methods, often hoping for results that rarely came out as planned. Then, researchers discovered a natural system in bacteria that would unlock a new era in biology. CRISPR-Cas9, as it's called, allows scientists to target specific sequences of DNA, cut them with precision, and even replace them with new genetic material. It's fast, versatile, and inexpensive, and scientists are already using it to try to tackle problems once thought impossible. Curing genetic diseases, improving crops, engineering microbes, and creating therapies that were unimaginable just a decade ago. At its heart, CRISPR is a story of discovery, innovation, and unexpected insight. We were inspired by bacteria that can remember viral invaders well enough that the next time the invader comes around, it will cut the virus's DNA to stop it. Scientists realized that this same system could be adapted to edit genes in more complex organisms, like humans, allowing them to correct the genetic typos that cause disease. In this episode, we'll trace the path from the earliest discoveries to CRISPR's rise as a revolutionary gene editing tool. We'll unpack the science behind it, explore how it's being used in the medical and agriculture, and see why it has captured the attention of scientists, ethicists, and the public alike. By the end, you'll understand not just what CRISPR is, but why it matters, and how it is reshaping our understanding of life and our future. Before CRISPR became a household name for geneticists, scientists struggled to manipulate DNA with any real precision. In the 80s and 90s, researchers experimented with methods like zinc finger nucleases and TLAMs. These tools could target specific genes, but the process was slow, expensive, and unreliable. Editing even a single gene could take months or years, with no guarantee of success. The dream of precise, accessible gene editing just felt out of reach. Then came a surprising source of inspiration: bacteria. These tiny organisms, which have been living on Earth for billions of years, have evolved their own defense systems to survive attacks from viruses. When a virus injects its DNA into a bacterial cell, some bacteria capture snippets of that DNA and store them in a special region of itself called CRISPR, short for clustered, regularly interspaced, short palindromic repeats. Later, if the virus attacks again, the bacteria uses this stored DNA as a guide to cut the viral DNA apart. It's a natural immune system for these bacteria, and one that has been silently impacting us for our entirety of human existence. In 2012, two scientists, Jennifer Doudna and Emmanuel Charpentier, realized that this bacterial system can be adapted for use in labs. They showed that by designing a guide RNA, they could direct a specific protein called Cas9, basically molecular scissors, to a precise location in any DNA sequence. Simply put, they proved the existence of GPS system for a tiny scissors to cut DNA and change it. This wasn't just a small step, it was a leap forward. Suddenly, fast, precise, and reliable DNA editing was no longer impossible. In fact, it was now probable. The impact was immediate. Researchers could now experiment with genes in ways previously impossible, from modifying yeast in plants to correcting genes in even human cells. Labs around the world began testing CRISPR in a wide range of applications, and within just a few years, the technology had exploded into a global phenomenon. What made CRISPR so revolutionary wasn't just its precision, it was its accessibility. Unlike earlier methods that required specialized equipment and years of training, CRISPR could be done with far less capital and far less skill. It could even be utilized in a standard molecular biology lab. This opened the door for thousands of experiments, rapid innovation, and applications that stretched far beyond what anyone had imagined. Now that we've seen how CRISPR was discovered, let's dive into how it actually works. At first glance, the idea of editing DNA might sound like science fiction, but the mechanism is surprisingly elegant. CRISPR relies on two main components: Cas9, the protein that acts like molecular scissors, and the guide RNA, the molecular GPS system. Simply put, Cas9 is the tool that does the cutting, and the guide RNA is the map that directs it to the right spot in the genome. Here's how it works step by step. First, scientists design a guide RNA that matches the DNA sequence they want to edit. Think of it like programming a GPS coordinate. Next, the Cas9 protein binds to the guide RNA. Together, they move through the cell's DNA until the guide RNA finds the exact matching sequence that it's looking for. Once it finds the target, Cas9 cuts the DNA at that exact location. After the cut, the cell tries to repair the break on its own. This is where things get interesting. There are two main ways that the DNA can be repaired. First, non-homologous end joining, or NHEJ. The cell simply stitches the DNA back together. This method is quick, but it could introduce small mistakes, which can disrupt the genome. These small mistakes come from the fact that there is now a missing link section in the genome, the part cut out by the Cas9. Second is homology-directed repair, or HDR. If scientists provide a piece of DNA as a template, the cell can use it to replace the cut section accurately. This allows them to insert new genetic material, effectively rewriting the gene. But there's more happening at the molecular level than just cutting or repairing. Cas9 doesn't act alone. It requires a small sequence in the DNA next to the target site, called a PAM sequence, a protospacer adjacent motif. Without this tiny sequence, Cas9 won't recognize or bind to the DNA. Simply put, the PAM acts like a lock that only the guide RNA can open. This helps ensure that the scissors don't cut random parts of the genome. Another fascinating aspect is the structure of Cas9 itself. The protein has multiple domains that perform distinct functions. One domain scans DNA, another unwinds the double helix to expose the target, and the third carries out the actual cut. Each part works in perfect coordination, like a tiny assembly line in the cell, ensuring precision and efficiency. The guide RNA also has two roles. The first is the spacer sequence, which matches the target DNA. The second is a scaffold that binds Cas9 and helps stabilize the whole complex. It is a GPS, as well as a handle for the scissors. Cas9 just cannot function without it. Finally, the cell's repair machinery is more than just a passive patch. Cells have to detect the break, recruit repair proteins, and actively coordinate the repair process. Scientists have learned to influence this machinery to favor one type of repair over another, either NHEJ or homology-directed repair. This increases the likelihood that the desired repair takes place, and therefore the desired edit takes place. Again, simply put, CRISPR is not just cut and fix. It's a highly coordinated molecular system involving recognition, unwinding, cutting, and controlled repair. This level of precision and control is what makes the technology powerful and versatile. But why is this such a breakthrough? Again, before CRISPR, editing genes was slow, expensive, and unpredictable. With CRISPR, scientists have much more gene editing power. To put it in perspective, imagine a book with millions of pages. Each page represents a stretch of DNA. Previously, editing a single word could take years, and there was a high risk of making mistakes elsewhere in the book. CRISPR allows scientists to go directly to the word they want, to change and rewrite it almost perfectly, like upgrading from scratching out words in a printed book to a script that edits a book on your computer instantly. This precision, combined with its relative simplicity, is what makes CRISPR revolutionary. When you hear about CRISPR breakthroughs in the news, it might sound as if science can just drop the Cas9 protein and its guide RNA into the body and edit out the DNA. In fact, that might be what you're thinking right now. The reality is far more complicated. Getting CRISPR's machinery into the right cells at the right time is one of the hardest challenges in gene editing. Cells aren't wide open doors. Their membranes act as protective barriers, and the immune system is constantly on guard for intruders. To get around this, scientists have developed several delivery methods. First, viral vectors. Scientists reprogram harmless viruses, using them as tiny couriers to carry CRISPR components inside cells. Viruses are naturally good at inserting genetic material, but they can sometimes trigger immune reactions, and it's tricky to control exactly where they go. Second is lipid nanoparticles. These are small bubbles made of fat molecules. Because cell membranes are also made of lipids, the nanoparticles can merge with them and release their cargo. This approach has already been proven in mRNA vaccines and is now being adapted for CRISPR. Finally is electroporation. In lab settings, scientists can use a jolt of electricity to temporarily open pores in cell membranes. allowing Cas9 and guide RNA to slip through. This isn't practical inside the body, but it works well for editing cells that are later transported into patients. Each of these techniques has its own benefits and downsides, but currently, electroporation and lipid nanoparticles are the leading methods of delivery, depending on if the genetic modification is in or out of the body. Even though CRISPR is remarkably precise, it isn't flawless. Cas9 works by pairing a guide RNA with a DNA sequence, but sometimes similar sequences elsewhere in the genome get cut by mistake. These off-target effects can have serious consequences. The biggest issues tend to appear in medical applications, especially when editing human cells for therapy. For example, if CRISPR accidentally disrupts a tumor suppressor gene, it could increase the risk of cancer. If it cuts near genes involved in the immune system, it might trigger harmful immune reactions. In stem cell editing, off-target mutations could silently persist and then multiply as the cells divide, making them especially dangerous for transplants. Even in agriculture, off-target effects can create unintended traits, like reduced yield or susceptibility to disease, which might only appear generations later. To address these risks, researchers are engineering high-fidelity Cas9 proteins that cuss less often in near matches, and they're using computational tools to design safer guide RNAs. These refinements don't eliminate off-target effects completely, but they're steadily reducing the danger. However, these high-fidelity proteins lack cost efficiency and time efficiency, taking much longer to make than their edits. Now, let's look at how scientists are putting it into use today. From medicine to agriculture, and even industrial biotechnology, CRISPR is no longer just a lab curiosity. It's actively shaping our world around us. One of the most exciting areas is human health. CRISPR has already been used to target diseases caused by a single faulty gene. These are called monogenetic disorders. For example, sickle-cell disease, which affects millions worldwide, is caused by one single mutation in a gene that produces hemoglobin. Scientists have taken stem cells from patients, used CRISPR to correct the mutation, and then returned the cells into the patient's body. Early clinical trials show remarkable results. Some patients have seen their diseases vanish entirely, and in some cases, they've achieved what doctors call functional cures, where the body behaves as if the disease never existed at all. Inherited blindness is another area where CRISPR is making a real difference. Certain forms of blindness occur when a single gene fails to function correctly. Using CRISPR, researchers can precisely edit these genes in individual eye cells, potentially restoring sight. In early trials, some patients have regained partial vision, giving them the ability to perceive light and shapes again. It's a tangible example of how CRISPR is turning what was once impossible into reality. Cancer research is also being revolutionized by CRISPR. Scientists can engineer immune cells, such as T cells, to recognize and attack tumors more effectively. By editing genes that suppress the immune responses to T cells, these modified cells can target cancer with greater precision. While most of these therapies are still in clinical trials, the early results are promising, offering hope for patients with cancers that were once considered untreatable. Beyond these high-profile examples, researchers are exploring CRISPR for hundreds of thousands of other medical applications. From repairing mutations that cause cystic fibrosis to targeting viruses like HIV. The key is that CRISPR allows for precise, predictable edits, opening doors that conventional therapies cannot reach. Unfortunately, at the moment we are unable to target disorders that are caused by more than one gene. These are called polygenetic disorders. They encompass many of the most common and most problematic disorders humans suffer from, things like diabetes or schizophrenia. The reason we are currently unable to target polygenetic disorders is that these disorders require multiple changes to the genome to repair. Furthermore, we can't just use the Cas9 protein multiple times. It's more complicated than that. Too many cuts in the DNA causes stress. It could break the strand, causing potentially bigger issues than the disorder itself. Additionally, when targeting more than one gene, potentially up to dozens, it becomes much more likely to have a modification that isn't precisely correct, which would also cause issues that could be worse than the original disorder. CRISPR is also transforming food production in remarkable ways. Crops can be edited to survive drought, resist pests, or tolerate harsh soil conditions. For example, scientists have developed rice and wheat varieties that can withstand extended dry periods, which could be vital as climate change intensifies and a greater ratio of fresh water is needed to be allocated to human drinking water. Additionally, tomatoes, mushrooms, and other vegetables are being engineered to resist diseases without chemical pesticides, making farming more efficient and environmentally friendly. Livestock is another frontier. Researchers are experimenting with CRISPR to create animals that are more disease-resistant or grow more efficiently. Pigs, for instance, have been edited to resist certain viral infections that previously devastated herds. Cattle have been modified to improve heat tolerance or reduce the risk of inherited disorders. These changes can make animal farming more sustainable and reduce the need for antibiotics and other interventions. One big issue with this, though, is the ethical implications. These livestock modifications only further our ability to mistreat animals and worsen their conditions. We'll go deeper into this topic later. CRISPR is also revolutionizing research beyond medicine and agriculture. In industrial biotechnology, microbes are being edited to produce biofuels, pharmaceuticals, and other valuable chemicals more efficiently. Yeast, bacteria, and even fungi can be programmed to perform complex chemical reactions, cutting production costs and reducing environmental impact. Environmental science is another emerging area. Some researchers are exploring CRISPR to engineer microbes that can break down pollutants or absorb carbon more efficiently. Others are investigating gene drives, controlled edits in insects like mosquitoes to reduce the spread of diseases such as malaria. While these applications are still experimental, they demonstrate the wide-ranging potential of this technology. Even in basic research, CRISPR has accelerated discovery. Scientists can model organisms with specific genetic changes to study diseases, test drugs, and explore fundamental biology in ways that were unimaginable a decade ago. It's a tool that speeds up research in virtually every branch of biology, from neuroscience to microbiology. Simply put, CRISPR is no longer just a breakthrough in theory. It's a breakthrough in practice. Its applications touch our health, the food we eat, and even the materials and chemicals we rely on every day. The technology is shaping not only the future of science, but the very fabric of everyday life. We will talk more about this topic in the second part, including the big question about how this could grow and impact in our future, what the implications and challenges are that we will have to go through throughout that growth process, and the ethical implications and opinions on CRISPR.
Thanks for tuning in to the Breakthrough broadcast. If today's conversation sparked a new idea, share the episode and follow the show wherever you listen. Got a question, idea, or breakthrough of your own? Reach out at breakthroughbroadcastconnect@gmail.com. Until next time, keep questioning, keep building, and don't wait for the future. Help create it.